new PDF. I'm going to do a bit of a review on what I had covered that did not get included in the exam number 2, right? So these things are going to be on your exam number 4. And we have some interesting topics to cover today. So we are going to review the eukaryotic transcription, what is important to know about that, and we're going to start telling the story on gene regulation of prokaryotes. Okay? And if it happens that we have time, we can start on eukaryotes, but most likely we're going to stay with the prokaryotes. In case you're curious, this is lunch. So, all right. So let's start here. How do you start your eukaryotic transcription, right? Well, first thing, remember, you have to distinguish what is replication, what is transcription, then what is translation, right? So replication, you're making new DNA. Transcription, you're making RNA from DNA. And with transcription, you're going to have also RNA, mRNA processing, right? And this is property of messenger RNA. Then you're going to have export to the cytoplasm, and then you're going to have translation in the cytoplasm. So the translation is going to be making protein out of your messenger RNA codes. Okay. So here, when you're talking about eukaryotic transcription initiation, you're going to have your transcription factors. Remember that you're going to have those assemblies, the assembly of your transcription factors before polymerase can join that promoter sequence, right? So you have your transcription factors that are going to assemble. Remember, you also have the Tata box in the eukaryotes, but then the Tata box is a bit further upstream in the eukaryotes than it was in the prokaryotes. So that is your Tata box. Now, what is going to bind to your Tata box? Well, that's going to be your transcription factor assembly, your transcription factor of proteins, right? Is a complex that the initial one is called transcription factor 2D. Okay, so that's the 2D. Is it just one protein? No, it's a bunch of proteins that get together. It's a complex. Okay, so transcription factor 2D has in the complex this protein that is called Tata binding protein. So that's the one that is going to recognize the consensus sequence that is the Tata box, right? And it's going to interact with it, and it's going to bend the DNA. Okay? So Tata binding protein is in that TF2D complex. So here you have, come on, it's not going. Why is not going? There you go. All right. So you have here your consensus sequence that is the Tata box right there, right? Consensus sequence is a stretch of DNA, nucleotides, right? That is conserved throughout the species, conserved region. Okay. And here comes your transcription factor 2D that has Tata binding protein in this complex of several proteins. Tata binding protein recognizes the Tata box, bends the DNA, which is not shown here, and is going to recruit other important transcription factors. So you get TF2A, you get TF2B, okay? And you're going to get the TF2F and TF2H. So those TF2F and TF2H, they're going to be very important for interaction of your RNA polymerase 2 with the initiation site. Okay? So TF2F is going to bring your RNA polymerase, and then TF2H, which is showing right here, okay, so that one is your TF2H, is going to open up a little stretch right after your Tata box, and is going to allow a bit of this stretch to be open in this RNA polymerase. So that's going to be your transcription bubble inside of the RNA polymerase, which is going to be like around here. Okay? So now what's going to happen? TF2H, besides having helicase activity, it has this very important phosphorylating activity. So TF2H is not only a helicase, but it is also a kinase. Okay? TF2H is going to add phosphate groups to the tail of your RNA polymerase 2. Okay? So very important property of TF2H. Okay? So here, what is happening? Once you have, this is important thing to remember, right? Once you have your tail phosphorylated, this is going to change the conformation of the protein stretch and is going to allow interaction. Yeah? You have a question? Sorry? You did this? No? Okay. So you have your tail phosphorylated, change a bit of the conformation. You're going to allow interaction with some other proteins. They're going to be very, very important for processing your pre-messenger RNA. So here are those proteins. Okay? The first one close to the end of the tail is your capping protein, capping factor. Then you have splicing factors, and then you have poly-A factors. And there is a reason that they are in this order, because this guy here is going to be the first one to hop out of the tail and interact with the nascent messenger RNA. So there is a reason for this order here, right? So this first one is going to interact with the messenger RNA. Remember, the first part of the messenger RNA that is being made is the 5' end. Why? Because all polymerases make polymers 5' to 3'. So this is going to be 5' coming out, then 3' is going to be inside. It's going to be 5' to 3'. Okay? So the capping factor is going to jump in there first. It's going to put the cap on that nascent RNA. Okay? All right. So here, let's see what's important. I just talked about that. This is the first one. The capping factor goes there and helps to put the cap on the messenger RNA. So what is important for processing of the messenger RNA? If the mRNA does not get processed, it will get destroyed and it will not leave the nucleus. Okay? If it's not processed properly, it will be destroyed. Right. So first thing that's going to happen is the capping. Second thing that is going to happen is going to be splicing. And the third thing that's going to happen is going to be addition of the poly-A tail. Okay? So you need to know the important parts of those processes. Okay? So here, we have seen this before, but just in case you did not pay attention to it because it wasn't in last exam, right? It's going to be in your following exam. So first thing that's going to happen with the 5' capping is that you're going to have an enzyme that is going to get in there with this 5' end, 5' because the 5' is at the tail, right? It's going to get there to the 5' nucleotide and is going to chop off one of the phosphate. So the name of the enzyme is phosphatase, right there, it's phosphatase. It's going to remove one phosphate. Okay, so you end up with two phosphates at this 5' end here. So now what is going to happen? You're going to have another enzyme that is called guanyltransferase. And guanyltransferase is going to add this nucleotide right here. This nucleotide is methylguanosine, okay, methylguanosine right here. Now, this nucleotide here, look, it is not a deoxynucleotide. It's just a nucleotide. Because remember, when you're making RNA, you're not using deoxynucleotide. Deoxynucleotide is to make DNA. This guy here is what? A ribonucleotide, right? Not deoxyribo. So it has the two OHs right there. So now this guy here comes with only one phosphate, okay? So this is what you call GMP. So you have guanyl monophosphate, one phosphate. this enzyme that is called guanylyl transferase is going to transfer this monophosphate and connect it to the two phosphates that are already sitting there. So you're going to end up with this strange connection of nucleotides that is called 5' to 5' connection. And it has 3 phosphates in the middle. Okay? So the name of the enzyme is guanylyl transferase. Then after that, you're going to add a methyl group to that guanosine, and that's going to become methylguanosine. So you have a methyl transferase that gets in there and put a methyl group right there. Okay? So 3 things that are important for the formation of your cap. Now, we didn't even start talking about the proteins. Once you have this conformation and the methyl group that is added there, they start to attract other proteins that are going to interact with this 5' end, and those proteins are going to help to protect the 2 functions. Actually, they're going to help to protect the 5' from exonuclease activity, and they are going to help, after the RNA is totally processed, they are going to help to direct the messenger RNA to come out of the nucleus and go towards the cytoplasm. So the complex of protein that is going to get in there is also going to serve as a guide to find the nucleopore complex and get exported out of the nucleus. Okay? All right, so that was the story with the capping. Okay, we're going to touch a bit more on capping when we go to nuclear export. Okay, so now capping is done. So now you have to worry about cutting out the intruders. So you're going to worry about RNA processing that has to do with kicking out the intruders, okay? Removal of introns. So you have 2 ways in which you can remove introns. Now, when you're talking about removing introns, we're mainly talking about eukaryotic messenger RNA, right? Because remember, prokaryotes don't really have a lot of time to deal with intruders. So it's a different type of processing. Eukaryotes, you have those pieces of introns, they have to be removed. Okay? You want to end up with only exons for your translation. Okay? So introns have to be cut out. Okay. How many introns you have to cut out? Well, it depends on the gene. Some genes have just a few introns to deal with. Some other genes have like as many as 60 introns. They have to be cut out all at more or less the same time before RNA can get finally finished processing and exported. All right, so how is it that the introns are going to be cut out? So some introns do the cutting on their own. So this is called autoproteolytic process. Maybe I didn't write it down somewhere, but it makes sense because it's doing proteolysis. Actually, this is actually talking about removal of something in the messenger RNA. But the process that goes on here is a kind of a process that usually proteins do that. So this is one example when RNA behaves like a protein. It has enzymatic process. So it's autoenzymatic process. We call it autoproteolytic process. Okay. So where do you see autoproteolytic process? When you have introns that are either group 1 or group 2. Okay. They do the cutting on their own. No help of other proteins needed. Where do you find this type of introns? Mainly when you have ribosomal RNAs. Okay. So how is it that this is going to happen? You're going to have a molecule of guanosine that is like right there in like close to the end of this intron. Okay, so this molecule of guanosine is going to attack a phosphate at the end of this intron. Okay, so this is at one end, it's going to attack a phosphate on the other side and is going to dissociate that phosphate from the RNA, and then this process is going to bind, the second reaction is going to bind the 2 ends of the RNA and liberate this intron from the messenger RNA. Okay, so this happens on its own without help of other proteins. Group 1 and 2 type of introns. All right, so those are group 1 and 2. So now, when you are dealing with other types of introns, you can have complexes, you can have situations in which you need the help of other proteins to do the catalysis. So this is when you need to get the spliceosome involved. So the spliceosome is a group of proteins that is going to interact with the intron and with the end of the intron, that is junction here of your messenger RNA exon, and the proteins are going to do the catalysis. We're not going to go into a lot of details on how that works because that always gave me a headache, and I always end up forgetting it afterwards anyway. So what I want you to know is that if you have more complex type of splicing, you need the enzymes and you need the spliceosome, and you're gonna need these proteins to interact with your exon and intron in order to cut the intron out. I think I left a little bit of detail, but not too much. So let's see what you need to know about the spliceosome. You need to know that those are proteins, okay, catalytic proteins. Okay, so we have a lot of proteins in there. No, you don't need to know them. Okay, I mean 50 proteins that are working in this spliceosome. That's a lot of proteins. Okay. So what you're gonna have? You're gonna have some proteins that are called here. Okay, let me see. U proteins. Okay. So besides the proteins, the U proteins, you're also going to need the help of some small RNAs. You call those small nuclear RNAs. Okay, so let's see what happens here. Okay. You're going to have this small nuclear RNA getting together with a lot of proteins that are part of the spliceosome. Those are the U proteins. Okay, and this thing is going to make a complex. Okay, small nuclear RNA is going to get together with proteins, and they're going to make a small nuclear RNP complex. Okay, so RNA with protein. We call this, the whole name of this is small nuclear ribonucleoprotein. Okay, another name for that is SNRP. So I have a picture here showing that it's not Smurf, it's NERP. Okay, now this thing here, this SNRP complex, is going to use energy to cut out the intron. So you need ATP hydrolysis for this thing to work. Did you need ATP hydrolysis for autoproteolytic process? No. But this is not autoproteolytic. This is using this whole complex of SNRP. It's going to need energy. Okay, you're going to need ATP. Okay. All right, so that movie was not in the video, but I will put the direct link so you can go and watch it again if you need to. Okay. All right, so let's focus here some more, because this is a part that gets more interesting. What is going to happen here is that we're talking about removing of introns, which is fine, normal. But in eukaryotes, you also have a process that allows removal of exons. And now you're thinking, oh, aren't exons important for making the protein? So yes, but here you're going to see something different that diverges a little bit from that central dogma that you had one gene making one RNA making one protein. Remember the central dogma? You probably heard about it in like biology 101, something like that. You know what I'm talking about? Yeah, central dogma. Okay, so what is the central dogma? Is DNA gene makes RNA capped and polyA, and that makes a protein. Of course, protein is folded, right? That's your central dogma. Now we are talking about one gene making more than one RNA. It's like, so now we can get more than one protein from the same gene, okay? The different types of proteins that you get, those are called isoforms. So isoforms of proteins come from the same gene, okay? So now, how is that going to happen? When you're doing splicing of the introns, if you have this mechanism for this gene, you can also do some alternative splicing of the exons. So now let's see what happens. Here you have a gene. So gene has 4 exons. Your messenger RNA that here is getting processed has 4 exons. And now the introns are not there. But now you're going to do alternative splicing. And in one messenger RNA, you're going to have only 3 exons. There are 1, 2, 3. In the other messenger RNA, you're going to have 1, 2, and 4 exons. So this messenger RNA is going to give you one protein. This messenger RNA is going to give you another protein. Okay? So these 2 proteins, they are called isoforms. Okay? So example here of isoform is with tropomyosin gene. Okay? Tropomyosin is a type of protein that is important for muscle contraction. Remember tropomyosin? Everybody took AMP? No? Have you ever seen about troponin, tropomyosin, and those okay, so let's think that tropomyosin is very important for mechanism of muscle contraction. Okay? Contracting the muscles. All right. So this is telling you that you can have 10 different isoforms that are coming from the same gene. Okay? Each isoform is going to be important for a particular cell type. So you're going to have one isoform that is going to be important for skeletal muscle contraction. Those are the muscles that you can control just because you think about contracting the muscle, right? You have muscles that are smooth muscles. So those are the muscles that you cannot control and they work on their own. Smooth muscles like muscles in your gut that do that, spontaneous moving of food around, right? What do you call that, the kind of movement from your gut? I went blank now. What do you call that thing? It will come to me in a minute. But so it's not spontaneous. You cannot control the movement, right? Huh? Peristalsis. Yes, peristalsis. Okay. So peristaltic movement, those are smooth muscles, right? So you're going to have one type of tropomyosin for that. And you're going to have a different type of tropomyosin for your heart muscles that almost look like skeletal muscles, but you cannot control your heart muscle, right? So different type of proteins are going to come from the same gene. So this is telling you that because of differential splicing, you can get different proteins that are coming from the same gene. Okay? So one gene is going to give you different proteins. So here is your example for tropomyosin. Okay. So here you have one form of processed messenger RNA that is for voluntary smooth muscle, another one for smooth muscle, another one for brain messenger RNA. Oh, that's interesting. So you're going to have tropomyosin in your brain. Why would you need tropomyosin in the brain? You're going to flex your brain? No, not really. But you're going to have some movement of stuff in your brain that depends on this type of protein. Okay? So different proteins coming from the same gene. All right. So that's the story with the splicing. Okay? Everybody cool with splicing? Okay. All right. So now we go for the last process of the messenger RNA, which is addition of your poly-A tail. This is an enzymatic process that depends on protein. Okay? So this, what happens? You do not have a code in your DNA that is going to be a code for poly-A. There's no such a thing. Okay? So what is going to happen? You're going to transcribe the gene, and you're going to keep on transcribing way after your coding sequence for your protein finishes, right? And how long? Well, look at this. Up to or more than 1,000 nucleotides after the end of your code, right? And then what's going to happen? You're going to have this enzymatic process. So gene here, transcription happen, and you have here about 1,000 nucleotides that got transcribed into your pre-messenger RNA, right? But then there is a region here that was coded from the gene, and this region here has another consensus sequence that is very much conserved for all of the genes that are going to become protein. Okay, so all of the genes here have this consensus sequence that is Okay, so that's a consensus sequence that gets into a pre-RNA. This consensus sequence is going to attract a protein that is going to interact with it and is going to count some nucleotides downstream, and is going to cut that piece of RNA that is not going to be translated, that is not going to be used for your finally processed messenger RNA. Okay, so all those 1,000+ nucleotides that just got transcribed because they did, those are going to be cut out. Okay, so you're going to have this enzyme that is going to cut the excess RNA and is going to add a poly-A tail to this part that is downstream, about 30 nucleotides downstream from your consensus sequence. Too complex for my taste. Okay, so you see, I mean, you can go through the stage in the book and learn how they're interacting, which one is stimulating what, which one is coming where, but you know, it's just a waste of brain power. Okay, you have to save your neurons. Okay, because you have to cover a lot more stuff. So not that the thing is not very interesting. I don't think it's very interesting, but that's my bias. Okay, so what's going to happen here? First thing you need to do, you have to look at your transcript of pre-RNA and you say, I have a lot of RNA that is useless. I have to cut it out. So the first enzyme that is going to recognize the consensus sequence is going to cut out that piece of junk messenger RNA. So that enzyme is the one that's going to cut. So we call it cleavage something enzyme. Okay? Cleavage enzyme that is going to help with adding of your poly-A tail. So that's the name of the enzyme. Okay? After that, you're going to have to add the poly-A tail, and it's an enzymatic process. So if you have an enzymatic process that's going to add adenines, you're going to do some polymerization. So the name of the enzyme is some polymerase. Okay? What is it going to do? Add adenines. Oh, so it's several adenines, polyadenine. Polyadenylate polymerase. That's the name of the enzyme. You add how many adenines are going to be added? Anybody want to guess? Depends on the gene. You have sometimes a short tail, sometimes you have a long tail. There are advantages for having a long tail because that gives more stability and half-life of your processed messenger RNA. Okay? The bigger the tail, the longer the RNA will live in the cytosol. Okay? So it really depends on how big you need, how long you need the RNA to live. Okay? So that's the story with your poly-A tail. Everybody cool? So 3 processes: capping, splicing, poly-A tail. Okay? Capping, there is only one way to cap. Splicing, there are 2 ways to splice. Adding poly-A tail, there is only one way to add the tail. So now we're going to look at, so here for your ribosomal RNA, you're going to have intron removal, and you're going to have other modifications. And mainly what you're going to have here at the nucleolus, you're going to have assembly of your ribosomal RNA with some other proteins, and once your final assembly is made, remember our ribosomal RNA, you have a large subunit and you have a small subunit, right? Once your subunits are assembled, then those subunits are going to be exported to the cytoplasm. But the assembly is going to happen at the nucleolus. Okay, everybody cool with that? Okay, and look at that. It is red, it is bold, and it is underlined. That makes a good test question. Right? Am I telling you? Yeah? Yeah. Okay. All right. This is going to go no clue. So now some fun stuff about transcription. you have some very powerful inhibitors of transcription. All right. So here you have this weird looking protein. How do you know it's a protein? Well, you should see some peptide bonds here if you kind of look for them, right? So this is a weird looking protein, right? So this thing is called actinomycin. Actually, this thing is actinomycin D, okay? So what happens here? You have some analogs of nucleotides, specific ribonucleotides, right? Because if it was deoxyribonucleotide analog, that would interfere with making of DNA. So these guys are interfering with making RNA. So those are going to be analogs of ribonucleotides. Okay? What are they going to do? They're going to interfere with making RNA, of course, right? So they're going to stop RNA polymerase, mainly because these analogs don't have that leaving group that is needed to continue the chain. Same story as we have seen with inhibitors of replication. Right? Do you remember the name of that inhibitor of replication that was on the test? Chemotherapeutic agent? That's fine, I forgot it. So here, RSC or something like that. Huh? Acyclovir, all right. RSC is another one, acyclovir. All right, so here, actinomycin D, right? So actinomycin D was actually one of the first antibiotics used, but it's so toxic, and then derivatives of actinomycin was very toxic. Now actinomycin D is also used as an antibiotic because it inhibits transcription in bacteria, right? So you can extract actinomycin D from some fungi, right? And you can stop replication of bacteria by inhibiting the transcription in the bacteria. Now, actinomycin, it is toxic to us because it's not very specific for bacterial transcription. Actinomycin D is a bit more specific for bacterial transcription, so it might not affect us eukaryotes as much. It's still a bit toxic, but you know, the poison is always in most of the time is in the dosage. But then you have some other poisons of transcription that are very, very effective and powerful to stop eukaryotic transcription. So here's what we're going to see next, right? Death cap mushroom. Who heard of this thing? 2 people out of everybody in the class. Okay, let me ask again. Everybody's awake? Yeah. Have you heard the story that you should not go around picking up mushroom and eating them? Thank goodness. Yeah. Why? Because look at this guy. It looks like those fancy big mushrooms that you see in the supermarkets, doesn't it? What is the name of the big mushroom? Portobello. Yeah. Okay, my brain is going blank. You know, I'll blame. See, I can't forget my word. Perimenopause. Yay. Okay. So sometimes I brain goes blank. Oh, so portobello mushrooms. No, you don't go out harvesting your own portobello mushrooms, because if you pick up this guy that is a death cap mushroom, what's going to happen to you? You're going to die. You're going to die. There is no antidote for death cap mushroom. Okay? It is deadly. Okay? Why? Because it's going to stop transcription. Okay. Delicious, yeah. And then somebody asked me, oh, aren't all mushrooms edible? Yeah, they are. Some of them only once. It's true, right? Okay. Hey, that's a joke, okay? They're not edible. They're not all edible. All right. So what happens here? If you have your death cap mushroom, you can extract this protein. Look at it. It's a protein. It is a protein. Talk about a deadly high protein diet. Okay, so this thing is called alpha amanitin, okay? Look at this, the deadliest of all of the amatoxins. So that's a toxin, okay? What does it do? It is going to block RNA polymerase 2 and RNA polymerase 3. So you're not going to get messenger RNA. You're not going to get transfer RNA. You might get some ribosomal RNA, but if you don't have the other two, you cannot make protein anyway. Okay? Very, very deadly. Okay. And I did put a link there for one case that I believe was in Australia a couple of years ago. There was a case of this woman that decided to get rid of her in-laws. Anybody heard of that one? No? Yeah, you heard of that story? And she ended up poisoning her own son. Yeah. Very sick story. Anyway, there's a link for that in case you're wondering, you know, if anybody ever uses this. But anyway, she was caught. I think she is still in jail like forever. So here, I put 3 slides for summary of your transcription. Okay, let's focus. I still got time. Okay, we're still gonna go to prokaryotes. All right. So these slides concentrated with the most important parts that you have to know about transcription. Okay, that's your study guide for transcription. Got it? Yeah. Make sure you get those 3 slides, put it into a separate PDF, dump it into ChatGPT, and ask ChatGPT to come up with 50 multiple choice questions that I could possibly ask you on a test. That's a good way to study it. Okay? All right. So now, any questions about transcription anymore? We're done with transcription. Finally going to go to regulation of gene expression. That's chapter 7. We are going to start with the prokaryotes. Okay? How it is that prokaryotes regulate their gene expression. Then we're going to go into eukaryotic regulation. Okay? But we're not even going to start talking about eukaryotes today. Focus on whatever we have time left for prokaryotes. 20 minutes. Okay? On the prokaryotes. Then other very much fun stuff. Okay. So, why is it you need regulation of gene expression? Well, as I mentioned before, you don't want all of the cells to be expressing all of the genes like all of the time, because that's waste of energy, right? So you have to have controlled gene expression. Here we have an example that in case of eukaryotes, you have certain differentiated, terminally differentiated cells that are supposed to express certain genes. Other terminally differentiated cells express other genes, and depends on the function of the cell. Like, for example, in your brain, you don't want to have the same proteins that are expressed in your liver, for example, right? But the DNA of both of the cells, the DNA is the same, but you have to control the gene expression. So now when you start talking about prokaryotic control, remember prokaryotes don't have that nuclear membrane, right? So prokaryotes have to do all this control of gene expression in one space that is the same type of space that the thing is going to do translation into protein. So it has to use different strategies when you compare to eukaryotic strategies. Okay, it doesn't have a lot of room to control this stuff. First type of control that the prokaryotes have, you already seen it, is the presence of sigma factors. If you have a certain sigma factor, you upregulate a certain gene, right? Now, remember the sigma factor is the thing that chooses the gene, and then the core protein does the rest of the job. We've already seen that. All right. So now we're going to look at operons. Yes, they tend to be a nightmare, but they're not that bad. We're going to make it really, really simple, okay? You have to know about 2 types of operons. The first one is going to be inducible operon. The second type is going to be repressible operon. Now, telling you the story, does it make sense from the bacteria perspective? Okay, so bacteria is sitting there and saying, I have to control my gene expression. I can either upregulate or downregulate my gene expression. So I can have inducible or repressible. Those are the two types. Okay. So first thing that we're going to look is by the use of operon. That's how bacteria control the expression by operons. Okay. What is in the operon? Here. What is in the operon? You're going to have a promoter region. Okay. Even before the promoter, you're going to have another region. Look at this. You're going to have a region that is going to make a regulatory gene expression. So this other region here has its own little promoter that is always on. Okay, so you're going to make this regulatory gene. You got it. Okay, regulatory gene. Okay, before the rest of the promoter, before the rest of the operon. Okay, this is important part of your operon with its own little production site. Okay, then you're going to have here this promoter region. Now it makes sense for this thing to be regulated at the promoter level, right? Because right after the promoter, you're going to have all of the other genes in tandem sequence. So you're going to have gene 1, gene 2, gene 3, gene 4. If your promoter is free to bind your RNA polymerase, remember only one for bacteria. If the promoter is free to bind RNA polymerase and you turn this thing on, then all of the genes are turned on. If the thing is off, then all of the genes are turned off. So this is a case of all or none type of gene expression. Now look at this. This is gene 1 is going to make one protein. Gene 2 is going to make another protein. Gene 3 so forth. Okay? So what is in the operon? You have a regulatory gene expression, promoter region, operator region that is part of your promoter, and then you're going to have your genes. So that is your operon. Okay, so let's look here at your inducible operon. You're going to have only one example of inducible operon, and that is your lac operon. Anybody heard of the lac operon before? Yeah. So you know everything about the lac operon? Okay, there was a silence. All right, so here is what you need to know about the lac operon, right? Usually, bacteria like to eat glucose, okay? Sometimes bacteria doesn't have glucose, it likes to eat lactose, whatever it has available, right? So this is lac for lactose, right? So lactose is a different type of sugar. So here, let's say that if you don't have lactose available, you don't want to turn on the genes that are going to allow you to digest lactose because that's going to be waste, right? If you have glucose available, you don't want to turn on the genes that would do lactose digesting because you already have glucose, right? If you have glucose and lactose, you still don't want to turn on the operon because you got plenty of glucose, right? So let's see how this thing is controlled. So first thing that is going to happen here with this operon is that you are going to have this regulatory region that is going to produce a little protein that is going to be your repressor protein. So look what's happening here. This is your gene. The name of your regulatory gene here is LAC-I, I for inhibitor. Okay, so LAC-I gene makes your messenger RNA. Messenger RNA makes your protein that is your inhibitor, or also called your repressor. The repressor protein goes to the operator region in your promoter and binds to it. Okay, it binds to it because it doesn't want you to be making genes that you're not supposed to if you don't have, if you don't want to digest lactose. Okay, so this repressor is always interacting here with the operator region of your promoter. If the repressor is there, then RNA polymerase cannot bind to the promoter region and you cannot make your gene products. Okay, so repressor is there, and how does it get there? How does it get made? It's made before all this whole story of control. Okay, so repressor is always made. Then this is the case that, let's say that you have plenty of glucose and you don't have any lactose around. Now let's say that you run out of glucose and you have lactose around. Okay, so what is going to happen here? Well, here is telling you the story that what happened if you have lactose around. First thing that's going to happen, oh, it is missing a part of the story here. Okay, so let me add the part of the story that is missing, okay? So here is your cell, so this is your bacteria, okay? So here you have lactose around here. Lactose is sugar, right? And here is the inside of the cell. So here's the cell DNA, okay? Now, in order for you to get lactose inside of the cell, you need to have a channel that is going to go right there. This is a channel. And it's going to allow lactose to get inside of the cell. Lactose is not going to just cross the membrane, right? So the gene that is going to make that channel is a gene that is called permease. Now, if you look here at your operon, where is a permease? It's right here. What is the gene that makes permease? It's the LAC1, okay? So you're going to have to make permease to put this protein over there to allow lactose to get inside of the cell. So now this is a part that's a bit confusing because we just said that without lactose, this thing is always off, like this. But we are saying that you need permease to be expressed in order for this channel to be in the membrane and to get lactose inside of the cell. So how can the two things be happening at the same time? How can you have this thing off but be on at the same time? What's going on here? So this is the part that tells me that when you learn about the lac operon, it wasn't very well emphasized. Huh? Linkage transcription, yes. Oh, let me see. Yes. Ready? Ready? Yep. Yeah, almost. Linkage transcription. What does that mean? It means that this guy here that is repressing the operon, it's not there all the time with crazy glue. Okay? This sometimes it falls off. But most of the time, 99.9999%, it is bound to the operator. But a few times when this thing falls off, then polymerase can go and make some of those RNAs. And you get a little bit of protein. It's like a very minimal amount of protein out of this operon that is mostly shut down. Okay? So you have a little bit of transcription that goes on, even if the operon is off. Okay? That little bit of transcription allows for a little bit of permease to be there at the membrane and to allow some of the lactose to get inside of the cytoplasm if you have lactose outside of the cell, in the environment. Okay? So this is one idea that is important to have in mind. Okay? So once you have lactose inside of the cell, you have another enzyme that is going to transform lactose into allolactose. So you have here galactosidase, transacetylase, now I don't remember which one is which, but one of them is going to get the lactose and make this other protein that is called, this other sugar that is called allolactose. Allolactose is able to bind to your repressor and pluck it out of your promoter. Okay? So allolactose goes there, plucks out the repressor, and now your polymerase can make your transcripts, right? So you need to have lactose inside of the cytoplasm in order for you to open up the operon and to start to activate your operon. This is why we call this inducible operon. It's not on all the time. Okay? There is more to this story. Okay? So this thing here is your inducer. The thing that plucks out your repressor, that is your inducer. Everybody got it? Okay. All right. So here, yes, we talk about that. Okay. So more details about the lac operon. So you have two-part switch. You have to take into consideration the presence of glucose and the presence of lactose. Okay. So now what is going to happen? Let's see here. Remember that you have a little bit of expression that goes on. Okay, I just mentioned that. No pictures in this slide, so you can read that on your own. Okay, so now the second part. Not only you need to have lactose presence, you also need to have absence of glucose. Because if you have both, then the cell will prefer to use glucose. Okay? Now, if you are using glucose, you are going to have low levels of cyclic AMP. Okay? So a lot of glucose means that your cyclic AMP level is down. Okay? Let's not go into the biochemistry of how that happens. Just keep that association in your mind. Okay? So if you have glucose present, you have very little cyclic AMP. Okay. If you don't have glucose, your level of cyclic AMP goes up. That's the first thing you have to remember. Okay? Now, what is going to happen? With elevated levels of cyclic AMP, that cyclic AMP is going to bind to a protein that is called activator protein. Cyclic AMP activator protein, okay? This activator protein will need to go and bind to the DNA and allow activation of your lac operon, okay? Without the activator protein, your RNA polymerase is not going to do a good job, okay? So let's see how that is happening here. In order to have activation of your cap, you are going to need to have a higher concentration of cyclic AMP to get the cap activation. Okay, so here is your cap protein. Okay? Without cap protein, you are not going to have polymerization, RNA polymerase to do its job. Cap protein is the activating protein that is going to allow polymerization. How are you going to get cap to interact with your DNA? You're going to need cyclic AMP. So let's put some cyclic AMP right here. Cap is going to be bound by cyclic AMP right there. Okay, so this guy here is, okay, let's put it here actually. If your cap is interacting with the DNA, it is bound to cyclic AMP. Okay, cyclic AMP. It's there, it's here. If you do not have lactose around, your repressor is also there. So binding of cap with cyclic AMP, meaning that you already don't have glucose, but you also don't have lactose, is not going to upregulate your lac operon. Most likely the cell is going to figure out something else to eat. Okay? Now, if you have lactose, so your repressor is not there, you don't have glucose, so now you have high level of cyclic AMP, your cap is there, now your RNA polymerase can go to the promoter and work properly. Okay? So two things you need. You need presence of lactose, absence of glucose, which means higher level of cyclic AMP. Okay? All right. So here is a little summary of how this is working. Okay, you have to know what is happening for each one of those scenarios. When your operon is on, when your operon is off, how to turn it on, how to turn it off. So basically what I taught here, that's what you need to know. Okay. So now let's, oh, finally, I knew I had to put some. Get your iClickers ready. Okay. So this is the time when you can consult your classmates to get the correct answer. All right. The LAC-I gene produces which of the following? Okay. Right, 10 more seconds. Does anybody need more time? Yes, okay. Let's put in your answers. Everybody put in the answer? Yeah, everybody's good? Okay, I'm gonna stop it. What is your answer? A. The black I is for the inhibitor, it makes your repressor protein. A, repressor protein. Your inhibitor makes a repressor protein. Okay. All right. So we talk about your inducible operon. Now we're going to talk about repressible operon, and only one example of a repressible operon. And this is going to be your tryptophan operon. Okay? So what is the idea here? The idea here is that you need to make tryptophan in order to keep the cell alive. What is tryptophan? It's amino acid, right? So it's not an essential amino acid because this thing is making it, right? So you need to make tryptophan, and you need tryptophan to be always at a certain level. So you need this operon on most of the time. When you reach a certain level of tryptophan, you want to shut this thing down. Otherwise, you're going to have not a good balance of those amino acids, okay? And you're going to shut this thing down in two different ways. One way is that, look, the operons, remember what they are made of, right? You have your regulatory region here that has your own promoter. This thing is going to make your repressor. And then you're going to have here your promoter region with a site that is the operator and the promoter. The operator is going to bind the repressor. But in this case, the repressor is not bound to the operator. The repressor is only going to bind to the operator when you want to shut this thing down. This thing is going to be on if you don't have enough tryptophan in the system, okay? So here, this thing is making the different proteins, all the genes in tandem, blah, blah, blah. So now let's say that you have enough tryptophan, you want to shut this thing down. The presence of tryptophan is going to bind to your repressor. Now you activate your repressor to go at your operator region and block your polymerase. So that's the first level of repression of this repressible operon. Okay, everybody got this level? Yeah. Okay, so I know time is running, people are packing, and I'm just going to start talking a little bit about the other way of suppressing your operon. Okay, this is the part that gives a bit more of a headache. Okay. This part here, the part that is called attenuation. Attenuation always gives me a headache. Okay, so I'm going to start it now, and I'm going to wrap it up Friday. Okay, what is attenuation? It's folding of the messenger RNA. Depending on the folding of the messenger RNA, you're going to either allow polymerase to keep on polymerizing, or you're going to kick out the polymerase from the system. Everybody got that in your mind? Attenuation has to do with the shape of the messenger RNA. Okay, now I'm going to stop here. Do you have questions? No, like at this time, they're just fast and run. Got it? I got it.